Good morning, students. Our today's topic is the C4 pathway. Plants that are adapted to dry tropical regions have the C4 pathway. Tropical region means near the equator. Though these plants have the C4 oxalic acid as the first carbon dioxide fixation product, they use the C3 pathway or the Kelvin cycle as the main biosynthetic pathway. C4 प्लांट्स में जो फर्स्ट स्टेबल प्रोडक्ट बनता है वो एक C4 कार्बन है, दैट इज़ ऑक्सैलो एसिटिक एसिड। इन प्लांट्स में C3 साइकिल भी चलता है और शुगर का फॉर्मेशन सिर्फ C3 साइकिल में और केल्विन साइकिल में ही होता है, जिसे हम बायोसिंथेटिक पाथवे भी कहते हैं। C4 plants are special. They have a special type of leaf anatomy. They tolerate high temperature. They show a response to high light intensities. They lack a process called photorespiration. In photorespiration, it is a waste process. About this, we will study in the next topic. Photorespiration that occurs in C3 plants, it does not occur in C4 plants, and this gives because photorespiration is absent in them, so these plants give higher productivity of biomass. Again, in C4 plants, the first stable product is oxaloacetic acid. In them also sugar formation occurs in the C3 pathway or the Kelvin cycle. Now about the structure. The particularly large cells around the vascular bundles of the C4 pathway plants are called bundle sheath cells. Around the vascular bundles means xylem and phloem, bundle sheath is present. The leaves have such anatomy are known as Kranz anatomy. Kranz anatomy means wreath, and it is the reflection of the arrangement of cells. Around the vascular bundles, the many cells are present in the layers. So the vascular bundles are surrounded by the different cells. This is known as Kranz anatomy. The bundle sheath cells may form several layers around the vascular bundles. They are characterized by large number of chloroplasts, thick wall, impervious to gaseous exchange, and no intercellular spaces. Now, in the bundle sheath, C3 cycle is there. C3 cycle takes place in bundle sheath and C4 cycle takes place in mesophyll cells. In the bundle sheath, rubisco enzyme is in excess quantity. The bundle sheath cells are rich in rubisco enzyme. Rubisco means ribulose, bisphosphate, carboxylase, oxygenase enzyme. Rubisco help in the formation of sugar. But here PEP case enzyme is absent. PEP carboxylase or PEP case enzyme is absent in the bundle sheath. Similarly, in mesophyll cells, here C4 cycle occurs. It lacks rubisco, but here PEP case enzyme is present. So difference between the bundle sheath cells and mesophyll cells. In bundle sheath cells, C3 cycle occur. In mesophyll cells, C4 cycle occur. In bundle sheath, it is rich in rubisco. Mesophyll lacks rubisco. In bundle sheath, PEP case is absent. In mesophyll, PEP case is present. Now, what is the process of C4 cycle? Just see the figure 13.9, diagrammatic representation of Hatch and Slack pathway. Hatch and Slack, the name of the scientist who described this pathway in C4 plants. First, carbon dioxide from the atmosphere enter into the mesophyll cells. There phosphoenol pyruvate, one compound is present which is a 3-carbon molecule. It combines with carbon dioxide and form a C4 acid. C4 is the first stable compound that is formed, that is the oxaloacetic acid. Then some transformation occurs, means isomers are formed, malic acid or aspartic acid is formed, and they transport from the mesophyll cell to the bundle sheath. In the bundle sheath, again decarboxylation occurs. Decarboxylation means from here the carbon dioxide is released. This again goes in this carbon dioxide which is released, it goes into the Kelvin cycle, because Rubisco always is present here in excess quantity in the bundle sheath. So this carbon dioxide combines with Rubisco. Rubisco is 5 carbon compound, combines with carbon dioxide. 6 carbon unstable compound is formed, which then breaks into PGA. The same process of Kelvin cycle occurs. After release of carbon dioxide from the C4 acid, what is left? That is C3 acid is left. C3 acid again comes into the mesophyll cell, that is a pyruvic acid, that changes into phosphoenol pyruvate, that is a starting compound. Again कार्बन डाइऑक्साइड मेसोफिल सेल्स के अंदर एंटर करेगी, वो फिर फॉस्फोइनोल पायरुवेट वहाँ एक कंपाउंड है उसके साथ में कंबाइन करेगी. जो फॉस्फोइनोल पायरुवेट है दैट इज़ अ 3 कार्बन कंपाउंड, वो कार्बन डाइऑक्साइड के साथ मिलकर एक 4 कार्बन कंपाउंड बनाता है जिसका नाम है ऑक्सैलो एसिटिक एसिड. यहाँ पे इसका एक और आइसोमर बनता है, तो ऑक्सैलो एसिटिक एसिड चेंज कर जाती है मैलिक एसिड में या समटाइम्स एस्पार्टिक एसिड में. वो फिर वहाँ से ट्रांसफ़र होकर आ जाते हैं बंडल शीट में. बंडल शीट में उनकी डीकार्बोक्सिलेशन हो जाती है, कार्बन डाइऑक्साइड रिलीज़ हो गई, वो कार्बन डाइऑक्साइड केल्विन साइकिल में चली गई. उसके बाद जो बच गया वो C3 कंपाउंड बच गया. C3 वहाँ से बंडल शीट से ना कर फिर मेसोफिल सेल्स में एंटर कर जाएगा और अगेन फॉस्फोइनोल पायरुवेट को फॉर्म करेगा. The primary carbon dioxide acceptor is a 3 carbon molecule that is phosphoenol pyruvate and it is present in the mesophyll cells. The enzyme responsible for the fixation is PEP carboxylase or pepcase. In the mesophyll cells, enzyme pepcase is present which help in the fixation of carbon dioxide with the phosphoenol pyruvate. The C4 acid is oxaloacetic acid that is formed in the mesophyll cells. It then forms other 4 carbon compound like malic acid or aspartic acid in the mesophyll cells itself, which are transported to the bundle sheath. In the bundle sheath, these C4 acids are broken down to release carbon dioxide and a 3 carbon molecule as already explained. The 3 carbon molecule is transported back to the mesophyll where it is converted into phosphoenol pyruvate again, thus completing the cycle. This is the C4 cycle. The 3 carbon molecule is transported back to the mesophyll where it is converted into PEP again. The carbon dioxide released in the bundle sheath cells enter the C3 or the Kelvin pathway, a pathway common to all plants. As I have discussed that for the formation of sugar on C3 cycle or Kelvin cycle is required, whether it is a C3 plant or C4 plant, but sugar formation occurs by C3 pathway only. That is Kelvin cycle. The bundle sheath cells are rich in an enzyme Rubisco but lack pepcase. Thus, the basic pathway that results in the formation of sugar, the Kelvin pathway, is common to the C3 and C4 plants. Did you note that the Kelvin pathway occurs in all the mesophyll cells of the C3 plants? In the C4 plants, it does not take place in the mesophyll cells, but does so only in the bundle sheath cells. In the C4 plant जो Kelvin cycle है वो सिर्फ बंडल शीट में ही अकर होता है. Next is photorespiration. Let us try and understand one more process that creates an important difference between C3 and C4 plants, that is photorespiration. To understand photorespiration, we have to know a little bit more about the first step of Kelvin pathway, the first carbon dioxide fixation step. This is the reaction where RUBP combines with carbon dioxide to form two molecules of 3-PGA, phosphoglyceric acid, that is catalyzed by the enzyme Rubisco. Rubisco, that is the most abundant enzyme in the world, is characterized by the fact that its active site can bind to both carbon dioxide and oxygen, hence the name given, ribulose bisphosphate carboxylase. If more carbon dioxide is present, it will combine with the carbon dioxide. And oxygenase, if concentration of oxygen is more, then it will behave like oxygenase. Rubisco has a much greater affinity for carbon dioxide than for oxygen. Imagine what would happen if this were not so. This binding is competitive. This is the relative concentration of oxygen and carbon dioxide that determines which of the two will bind to the enzyme. If the concentration of carbon dioxide is more, then carbon dioxide will combine with RUBP. And if the concentration of oxygen is more, then it will combine with RUBP combined with oxygen. In C4 plants, photorespiration does not occur. This is because they have a mechanism that increases the concentration of oxygen at the enzyme site. Where is that enzyme site? In the bundle sheath. In the bundle sheath, Rubisco enzyme is present. So when carbon dioxide comes, Rubisco enzyme, so RUBP combines with carbon dioxide there. This takes place when the C4 acid from the mesophyll is broken down in the bundle sheath cells to release carbon dioxide. This results in increasing the intracellular concentration of carbon dioxide. So in the bundle sheath, the concentration of carbon dioxide increases, and their Rubisco enzyme also is present. So it combines with RUBP and 3-phosphoglyceric acid is formed, that is Kelvin cycle occurs there. In turn, this ensures that the Rubisco functions as a carboxylase, minimizing the oxygenase activity. In the bundle sheath, the RUBP combines with carbon dioxide and behave like carboxylase because the concentration of carbon dioxide is more. Now that you know that the C4 plants lack photorespiration, you probably can understand why productivity and yields are better in these plants, because no wastage of energy. So in C4 plants, photorespiration does not occur. They can form the glucose efficiently, and these plants show tolerance to high temperature also. So this is about photorespiration. I hope this is clear. Next we will continue. Thank you.